right, thank you so much for having me today. I do want to start out, I sometimes struggle taking my teaching hat off, so I apologize if it feels like I'm teaching, but I do want to start out thinking about heterozygotes. So if you remember back to your basic genetics course on our classic models of dominance or additive effects, can anyone tell me if the heterozygotes are truly different or not in this Punnett square? If we're dealing with the dominance trait, do we really think they're going to be different or not? They have that big A, so they're going to be dominant, right? And they'll be a certain color or whatever we're thinking about. General thought process with additive models as well sometimes, but my whole question is that really the case? Is our heterozygotes, does it depend on where you got your big A allele in this case? And so I do want to give a quick basic example of additive effects, particularly of course, this is going to be in a vacuum. I'm not going to be able to do it when considering environment. But if we have the A is our reference allele, and then we're just calling that our baseline, and then our T is our alternate allele, which adds 25 pounds, hypothetical effect. Okay, it'd be really great if we could find ones that were just like this. But an individual with a genotype of big A, or in this case, just a reference allele, 600 pounds. You add that T to it, you get 25 pounds. You add another T to it, you get 650 pounds. You get 50 pounds because you have two T alleles. Again, it doesn't work this simply in real life. But if we are working in a vacuum, but you think if you have one T allele, it didn't matter where it came from, you're getting that 25 pounds. But my question, I do want to talk about heterosis because it's another important thing, particularly in the population that I'm talking about. Heterosis helps us tremendously when it comes to crossbreeding. It has what they hypothesize as various causes, such as the dominance, overdominance, or epistasis. But ultimately, it's when those F1 generations outperform the average of their parents. But in the classic model of heterosis, directionality does not matter, and you'll see an example here. Which breeds the sire and which breeds the dam in the classic models, and I emphasize the classic models because things have changed, does not necessarily matter. So in the crossbreeding, I worked through a series of questions to kind of refine down my hypothesis. But my question started out as, are heterozygotes always equivalent no matter which breed or parent the alleles come from? That was my first question. What really got me on this trail was I worked under Dr. Jim Sanders and Dr. David Riley, and everyone at A&M had a big role in the reciprocal birth weight differences between Bos indicus and Bos taurus sires. So either you've experienced it or maybe read about it, but typically when we have Bos indicus on our Bos taurus animals, it's just a Brahman Angus cross, versus a Bos taurus animal on a Bos indicus dam, the birth weights tend to be a lot bigger in our Bos indicus sired animals, anywhere from 5 to 10 kilograms it can be. It is very large, and from a production standpoint, it can cause a lot of issues when it comes to dystocia of birth weights, particularly if you were breeding Bos indicus sires to your Bos taurus heifers, which I do not recommend. And the sex differences are even more pronounced. So, we typically think males are heavier than females. Well, in Bos indicus sired and on Bos taurus dams, males are much heavier than females. Females are still pretty heavy compared to the reciprocal cross, but the sex difference is pretty large. And so theoretically, these animals are 50% Angus, 50% Brahmin when they're born, right? So breed composition wise, they're the same exact, but why do we see such differences in them? And even when it comes to the more Bos indicus you have in the sire relative to the dam, the bigger birth weight differences you have. And so that's what really got me started on this. Does the allele of origin, or where the allele came from, affect whatever trait that we're talking about? And I do want to state, this is not a new question. This is not me by any means making up any sort of groundbreaking question. A lot of people have asked that, particularly when it comes to reciprocal birth weight differences. But what really drove me to investigate this was a paper out of Florida by Zays and colleagues. And they looked at breed of origin analysis in a Brangus population. So they have all different levels of Angus and Brahmin crosses all the way up to what we think of a typical Brangus composition. And their question was, does the allele of origin have an effect on various production traits? They focused on hot carcass weight and marbling. And ultimately, they did see that there is an allele of origin effect. They modeled 3 different models. They did overdominance, additive, and typical dominance because they were interested in heterosis. But breed of origin does help, and what they did is they ran a GWAS, and they found that if they accounted for the breed of origin of an allele, that it did help improve detection of a very slow side throughout the genome that are associated with Carcass traits that we typically don't find if we do not consider breed of origin. And so for a crossbred or a composite breed like Brangus, they found it was extremely helpful to incorporate breed of origin, because if you think about it, and I'll have some slides later, a Brangus chromosomes are a mosaic of Angus and Brahmin. Okay, after years of crossbreeding and finally getting to that composite, you know, we say 5/8, 3/8, or whatever it might be, there's still a mosaic of that Angus and Brahmin, just due to how crossing over works and how after many generations, we get these mosaic chromosomes. And they did find that allele origin matters, and they hypothesized that being able to integrate this breed of origin would be pretty useful for our composites or any crossbreeding system. So I do want to point this paper out because it really was one of the motivations of why I decided to look at this. But I looked at it in a different population, and so my analysis does differ a little bit. We have talked a lot about beef on dairy, but we do know that their population is growing. Almost 70% of the dairies in the United States use beef semen on their dairy animals. So the population is growing in regards to the ones that are the beef on dairy calves that we see in the feedlot. And they do outperform their purebred dairy counterparts. And our cow herd is getting smaller and smaller. And so they are really filling a gap. But I don't want to make that sound like a threat in regards to beef producers. There is, you know, a potential for us to consider how can we contribute our beef genetics and what beef genetics works best on our dairy cattle in order to really increase the performance of crossbred dairy animals. So they are pretty genetically distinct. I just pulled, this is chromosome 1 from, I think I did 200 beef animals and 200 dairy animals. This is their principal components analysis of their genetics. So the beef animals actually cluster. These are Angus, Hereford, and Charolais animals, but we see a lot of variation in our dairy animals. These were all Holstein. So they are pretty genetically distinct. They are theoretically both Bos Taurus, but they've been selected for very, very different things throughout their generations. And that's one of the things that I'm going to talk about. But my question for this data set was, do parental beef origin, so the beef sire versus the beef or dairy dam, influence genetic effects on specific SNPs on end products and beef on dairy crossbreds? So it's not quite like our Brangus study because I do only have one direction of F1s, which we'll talk about here in a second. But that was my question. Do beef origin alleles behave differently than dairy origin alleles? So this is kind of, I love my Punnett squares. I am a geneticist teacher after all, so I try to put them everywhere. But I like visuals. We have our beef sire and our dairy dam. Doesn't really matter where they go, but beef is blue and dairy is red. These two individuals are heterozygotes. And so in our classic model, we would assume that these two individuals are the same, right? They have a big A or they have an R and an A, reference and alternate. But this individual got the reference from the beef animal. This individual got the reference from the dairy animal. So are these alleles equal? That was my question, visualized. And so these were the main focus of my study, particularly because I only had one direction. But is there a difference here? Why there could be a difference, as I mentioned, they have very different purposes, right? Maybe not so much way back in the day, but nowadays they have really selected dairy animals to the extent that it was detrimental to other traits, but really selected for increased milk production, milk quality, udder scores, udder quality, real selection here. On the beef side, growth, feed efficiency. Feed efficiency is also important in the dairy, but very different focuses. And so their allele frequency and allele population has diverged just simply because of the selection that they have been subjected to. And so the same SNP I'm really focused on is not necessarily the SNP itself, but the SNPs around it. So a beef animal and a dairy animal may have the same exact SNP, but there are tons of regulatory SNPs throughout the genome that affect gene expression. I am more on the molecular side of things, and so I love thinking about this type of stuff. But that is my question. Do surrounding SNPs potentially affect which allele is selected or which allele is expressed more than the other, and does it have to do with the origin of said alleles? And so I do want to say effect doesn't always equal sequence, because even if base pairs are identical, neighboring SNPs or just how the genome is packaged or epigenetic effects could make one gene be expressed in one animal and a gene not be expressed in the other animal as much. So for my approach, I had about 630 beef-1 dairy animals. I had phenotypes and genotypes on the offspring. There were about 42 different sires across 3 different beef breeds within this population. I did not separate between, I was just focused on beef versus dairy. I hope to be able to break it down based upon breed type eventually. And so the great thing about beef-1 dairy is the record keeping. And so they know the dams, they know the sires, and they have tons and tons of data, particularly on these beef-1 dairy animals. And so I use pedigree information and phasing software to be able to determine which allele came from which parent. And since I had both sire genotypes as well as a huge reference of Holstein genotypes, I was able to properly phase for each offspring which allele came from the sire and which allele came from the dam. And so in this case, which allele came from the beef side and which allele came from the dairy side, which is very, very important for this type of analysis. Then I separated haplotypes. So haplotypes, the dairy chromosomes that that individual inherited and the beef chromosomes that that individual inherited. And so after filtering, I had about 13,000 SNPs, so not a whole lot, but I did have to do some pretty stringent filtering based on which SNPs did overlap between all these individuals. So here's an example of what it might look like. Here's a SNP, just sort of SNP ID. And then these are the various genotypes that an individual can be with coding and phased. So if you've ever looked at a genotype file, most of the time you see a backslash instead of a bar. But if you see bars, that means it's phased. So parental, in this case, the software that I use, the parental one is the allele that comes first. Maternal one is the allele that came second. And so if you have a 0-0, both alleles are the 0 variant, which is oftentimes the reference variant from the beef sire or the dairy dam. 0-1 is a 0 from the beef sire, 1 from the dairy dam. Then we have the opposite, the 1 from the beef sire, 0 from the dairy dam, and then 1-1, meaning they get both of those 1s from the dairy and the beef sire. Again, 0 indicates that reference allele, 1 indicates the alternate allele in my case. And so these heterozygotes are what's super, super important for this analysis, because they're opposite, okay? Got a 0 from the beef, 1 from the dam, but over here we got a 1 from a beef, 0 from the dam. And so the heterozygotes, both on the parent side and the offspring side, were super important for me, because this was my main focus, just because I did only have one directionality of this cross. And so then I ran 2 different models, my null model, my alternate model. Both models included a response variable, of course, phenotype. In this case, it was Carcass characteristics, which I'll show you all later. Fixed effects of sex as a covariate, and then a random effect of their genetic relationship matrix, because some of these animals were related, so that helped me control for that relatedness. My null model, if you ever looked at a genotype paper, it doesn't look exactly like what you've seen, or a GWAS paper, but this is basically a basic GWAS. And it says SNP combined because that's how it would typically be coded for in a genotype. You would have it either have 0, 1s, and 2s, depending on what program you're using. And so that model, my null model, had did not account for what allele was which. It was if that animal has that allele, or if that animal does not have allele, or if that animal has 2 copies of that allele. Versus my alternate model, which I am labeling as parent of origin effects, which I'll say why here in a second. This is a type of model you'll see in those more often. But I modeled them separately. So we got a SNP on the paternal, SNP maternal, and each of their effects. And so then I compared the 2 models to see, okay, is the null model better than the alternative model? Or if it's not, where are the differences in the alternative model? As I said, I do need to specify this. So parent of origin has to do with genomic imprinting. So there are certain genes throughout the genome that are only paternally expressed or maternally expressed. The best way to differentiate and to identify those is to have reciprocal crosses. So I would have a dairy sire beef dam and a beef sire dairy dam. I only have one directionality of those. So it is for this type of analysis and my analysis here, I cannot disentangle the effects from breed of origin effects versus paternal of origin effects. I did try to combat that a little bit by, once I identified any significant SNPs, I went into the reference genome to see if it was located in any known imprinting or parent of origin regions throughout the genome. But I do want to have a precursor to that I can't 100% disentangle that, but that's why you'll see sometimes I refer to it as parent of origin or breed of origin, but it's the same in my case because parent of origin is always going to be the beef sire and the dairy dam. And so here are my end products that I use: dressing percentage, hard carcass weight, carcass ribeye area, fat thickness, marbling score, carcass value, and I also put total average daily gain because of course, that's an important trait. So let's get in. I didn't find a whole lot of significant SNPs. I did not expect to based upon my SNP sample size as well as my animal sample size, but I did find some interesting results. I'm not going to read out the whole entire SNP number, but this is the SNP reference number, and this is the location within the chromosome. This is on chromosome 1. And so what I found was for this particular SNP, getting this particular allele, so just think of this A allele, getting that alternate allele from the beef side of things, added a 57 pounds on hard carcass weight. But if an animal was this person, or this cow right here, negative 9.6% or negative 9.67 pounds. And then of course, p values. So this is what I was hoping to find, some sort of indication that yes, getting this reference allele from the beef animal is way better than getting it from the dairy animal. And so this is this SNP. A little bit more information about this SNP. It's actually in the BTLA gene, which is involved in immune regulation and T cell inhibition, has a huge role in moderating immune response and preventing excessive inflammation. So I know that's not something we automatically jump to when it comes to heart carcass weight, but healthier animals tend to gain more weight. They're not losing, they're not sick animals, may not be eating as much, not going to have a final higher heart carcass weight. So that health status is probably pretty important. So that's why I often, sometimes when I hear people talk about their GWAS studies, they get disappointed because they're like, oh, this gene is in some gene that doesn't make any sense to me. Well, there's probably some biological function in there that it probably does make sense, but regrettably, it's not as obvious as, oh, this is literally the heart carcass gene weight locus 1, and that's why we're seeing this effect. For a marbling score, found a similar thing, but it was opposite. So for getting a beef allele, you got a negative 24 points, maternal allele 20 points. Not as small as a p-value, regrettably, but you can see that it's not always going to be the same direction. I didn't necessarily go in with the assumption that it was going to be the same direction, but in this case for marbling score, and again, these are Angus, Charolais, and Simmental sires. I did not break it up into which was which. But this SNP actually neighbors this PLEK-HB1 gene, which has been associated with milk fat yield in Holsteins. No other research in cattle. However, in pigs, they have found that it was differentially expressed in adipose tissue and ones with divergent fatty acid profiles, suggesting it does have a role in lipid storage. So my hypothesis here is that throughout our generation, or throughout generations of breeding of dairy allele, they are breeding towards more milk fat, right? And so perhaps they're influencing this gene, which also has an effect on fat deposition, which could be why we see a more positive effect when it comes to that dairy origin allele. So I didn't see any effects when it comes to dressing percentage or ribeye area. Now, and I did want to mention, I forgot to put this on that slide, for heart carcass weight, there was an associated positive effect of getting a dairy or a beef allele on overall carcass value, and a negative effect on having a dairy allele for carcass value, which makes sense. Higher carcass, we see the same trend when it comes to that SNP, when it comes to heart carcass weight, and carcass value are tied together. So it does make sense that you see the similar directions at that SNP. Did find one for carcass fat thickness. Again, the paternal allele actually had a negative effect, while the dairy allele had a positive effect. And then for total average daily gain, I don't have the SNP numbers up here, sorry about that. The beef had a higher effect on total average daily gain versus the maternal had a negative effect. But again, no significant effects for dressing percentage or ribeye area. And those were the main end products that I focused on for this study. So I say tentative conclusions because again, for a geneticist study, 630 animals is not great. I mean, it could be worse, but it definitely could be better. It could be higher. And I wish I did have more SNPs, but I'm not going to complain about the 13,000 that I was able to test. Parental of origin does matter, and I did not put this on the slides, but none of those locuses that I did identify were located in any known imprinting regions. That's not to say that it's not imprinting. But they weren't, because the cattle genome is not complete in regards to imprinted alleles. So it could well be that they're imprinted, and this is not really a dairy versus beef, which we'll see what I'll see on the next slide. So some locusts did see strong breed of origin effects, but I had 13,000 of them, and a good chunk of them did not. So it's not something that's super, super common. And I do believe that the effects probably go way beyond just the sequence. So it has to do with the regulatory variants that I think have been, we don't necessarily always focus on, but those regulatory variants outside of genes have a huge effect. And that's where we can really see the divergence between crossbred animals and breed of origin. So how could this be applied? This is a very, very basic example. And of course, it's not as simple as this. It never is. But potentially, what if you find a whole bunch of SNPs that do show a specific inheritance breed of origin pattern, and you're able to select for dairy dams that aren't going to contribute that detrimental allele, and you select for beef sires that are always going to contribute the positive allele. So if you go back to the Carcass traits, you select a sire that has a 1-1, he's always going to contribute that plus, I can't remember the number off the top of my head, 54, and you're never going to get that antagonistic effect from the dairy dam. Of course, it's never that simple. You're never going to select just a single SNP, but this is the very broad application that I see potentially being able to use if we can find SNPs that do have these high breed of origin effects. So what's next? I would love to be able to disentangle breed versus parent of origin effects. It's not going to be super possible here because I do need dairy sired animals on beef dams, which are not super common. So it's going to be pretty difficult to be able to disentangle these breeds effects. I do need to expand my genetic models. I just did the additive effects here just to keep it simple, but thinking of things like dominance and other things that contribute to heterosis is very important. And this was really just a preliminary study in regards to the small population that I did have. But what really, really drove me from this, again, I'm more on the molecular side of things, and I love gene expression. If you went to my talk yesterday, you'll know that I love gene expression. I would like to be able to integrate gene expression analyses to these type of breed of origin effects to potentially identify what is referred to as allele-specific expression. So theoretically, we typically think we have two alleles, right? And we have equal expression of each allele. Chromosome 1 has allele A, the other chromosome 1 has allele A, you get equal expression of both of those A's. But my focus, particularly with imprinting and breed of origin, is that I do believe there is allele-specific effects, and I have a better visualization here. So again, in typical diploid organisms, we have two pairs of chromosomes. Both the maternal and the paternal allele are usually expressed equally, unless we're talking about those known imprinted genes, or there are some other regions as well. But allele-specific expression happens when one allele is expressed more than the other. So if you can see here, these are going to be the same genes, but this allele is expressed a lot more than this allele. And it may be because of a SNP within the gene, but I really think it has to do with regulatory regions around the genes in front of it. And those regulatory regions potentially make a beef-sired allele be expressed more than the dairy-side allele, for example. So this is what really drove my interest. I love gene expression and being able to integrate it into things. And so I would love to be able to do this on a larger population. And again, if allele-specific expression is focused on a locus with marbling, for example, potentially the beef allele is being expressed more than the dairy allele for some reason. We could actually scientifically confirm that by using gene expression. It really provides that direct evidence for understanding those functional differences. And it's not just people on dairy. This was a unique population that I was able to work with. I think what the work that they did in Brangus animals is applicable to a lot more of our composite or crossbred animals. For example, this is a Beefmaster chromosome 1, just a random Beefmaster animal I pulled. And so you can see the heterozygosity of breeds throughout the genome. And I would like to see if there's breed of origin effects in various breeds for things like growth, carcass characteristics, fertility, and particularly adaptation. So we can actually, this is actually mapped from reference. It's called local ancestry analysis. And it's not just about beef on dairy. That was my main point here, is that I use this analysis within a beef on dairy population, but it can be expanded to a lot more of our composite animals. And really and truly, what I feel like we could do is select for breed-specific alleles within our crossbred populations. We've been doing this already, not necessarily on the chromosomal level. There was a study by Crum and colleagues I do believe in probably early 2020, maybe. I can't remember the year right off my head. But they basically said they looked at local interest remapping across generations. And at the very beginning, when we were forming these composites, because they looked at American composites, they really saw that the first initial selection was on those carcass traits and traits like that. And then, so what they saw is more Bos indicate or Bos Taurus influence throughout the chromosomes. And then eventually they started moving the other way, and we saw more Bos indicus components of the chromosome for adaptation. So it's something that we've already been doing, but I'd like to really nail it down to more on the chromosomal level. For example, could we do breed complementarity at the chromosome level? Could we look at a bull that has a potential, of course, crossing over does mess some of this up, but could we look at a bull on the chromosome level and say he has, chromosome 1 has this composition in regards to breeds. It's beneficial to have the Angus here and the Brahmin here, and he has that versus an animal that does not have that. And then can we then match it with a dam to have similar effects? And really, I say it's breeding on the chromosomal level. That's always been kind of my goal, is to be able to, we do a great job of breeding for, doing genetic improvement, using SNPs, using genome-wide associations, but really being able to look at the chromosomes of animals and be able to make matches in that regard. So that is, I call it breed complementarity, more for those composite breeds in regards to being able to, because if you look back, this animal is not a heterozygote across its whole entire chromosome in regards to the breeds we're talking about. He's homozygous Hereford here, homozygous Shorthorn here, but he does have some heterozygosity there. And so could we potentially use heterozygosity so we have a little bit of Hereford and Shorthorn overlap here? Is that more beneficial versus having being homozygous Shorthorn, for example, at this locus? So overall, why does this matter? I really think it could be integrated, as they said in the Brangus paper, it could be integrated to help us with selection accuracy, being able to understand where and the origin of these loci come from. With the different advantages each breed has, I think it's super important to be able to consider that, but this is more considering it literally on the chromosomal level. And really optimizing those crossbreeding strategies. It's really going to, I think, advance the understanding of heterosis. I refer to it as the classic models. We're changing our ideas of heterosis as more studies come out. And so really being able to understand how heterosis is working, even our more advanced composite breeds, and discovering those new genetic markers that we potentially wouldn't be able to see unless we integrate that breed of origin analysis into the study.